Hello, I'm Amy Ellison. I'm a lecturer and research fellow at Bangor University, and today I'd like to give you a bit of an overview of my recent research into tackling disease issues in aquaculture. So why do we care about aquaculture? Well, it's desperately needed to grow a sustainable source of protein for the increasing human population. We can no longer rely on wild capture fisheries because they are desperately overfished, and if not, then they're sort of at their maximum capacity. So we need to increase aquaculture production in the decades to come to feed humans. But there's a lot of issues with sustainability and how we can increase aquaculture development in a sustainable way. And there's various issues with this. For example, things like the feed ingredients that we use to feed fish are not particularly sustainable at the moment. Fish farms are often placed in natural environments that can cause issues with environmental degradation. There are issues with escaped farm fishes causing issues with wild populations. And for me, most interestingly, that there's a massive issue with disease in aquaculture. So this is, of course, a big economic problem for the industry itself. It's a multi-billion-dollar problem annually each year, and it can account for incredible economic losses within the industry. So here I just have an example of salmon lice. So salmon is probably, for the UK at least, a very important aquaculture species, and you can see that an incredible amount of money is used to actually control sea lice in salmon farms, and it's not only just the actual sort of costs of either trying to treat the fish themselves, but the production losses from diseased fish, things like mortality and reduced growth rates, is also an economic issue. But it's not just economics. Again, environmental sustainability is a big problem with disease. So we are forced to use pharmaceuticals in fish farms, and of course there's a risk of those then entering the natural environment. The use of antibiotic supplements in feeds can then escape and leach out into the natural environment. And there's a big worry about things like antibiotic resistance actually developing in sediments around fish farms. And of course, with these very intense disease outbreaks in fish farms, they can also then spill over into the wild populations. And there's a lot of kickback now in places where fish farming is very intense, where we're seeing increasing disease outbreaks around these farms in wild fish populations, which of course is a massive ecological issue. And then finally, there's an issue with public perception of fish farming. There's a concern that we need people to be eating more farm fish rather than eating wild captured fisheries, but of course they're beginning to see and think about fish welfare on these farms, and if we see these horrific images of diseased fish, then there could be a concern that the public aren't going to want to eat farmed fish. So a lot of my research has been looking at how fish farming practices may actually be increasing disease risks. And as a molecular parasitologist, I'm really looking at this at the sort of molecular level, particularly in terms of gene expression responses, of how fish are responding to infection under different aquaculture treatments. So one of the first things that I did as a postdoc in Cardiff was looking at how stocking density impacts fish's responses to diseases. So here we have a fairly simple experiment with tilapia, which is, although not very commonly farmed in Europe, is a very important food fish globally. And so we held and raised these tilapia under two different densities, which I'll just call for simplicity low density and high density. And then after a period of time on these densities, I then challenged these fish to a fungal pathogen called saprolegnia. And then after infection challenge, we would then take the tissues of infected and healthy fish under these two densities and look at how their genes are being expressed to see if we can see any differences as a result of the stocking density. And what we saw immediately when we challenged these fish is there was a massive difference in how susceptible these fish were to the disease, depending on their stocking density. And perhaps surprisingly, we saw in low density a massive increase in disease susceptibility, that the fish actually did a lot worse at the low density. Now, if you understand cichlids, this may be not so surprising, because they're actually a really territorial species. And at these sort of relatively low densities, they're trying to form territories that become very, very aggressive to one another in the tank. And this is probably increasing the sort of social stress, and that's having a knock-on effect in terms of their immunity. In contrast, at high densities, they can't form these sort of territorial behaviours, and therefore they seem to be more resistant to disease, which is quite interesting. So what was happening at the level of gene expression? Well, in both densities we kind of see a suppression of certain immune factors, and this isn't too surprising. We actually know that this fungal pathogen, saprolegnia, is actually able to produce these virulence factors that actually suppress the host's immune system, and we certainly were seeing that in both stocking densities. But what was interesting is in this sort of more stressed group, at low density, we see quite a large alter in gene expression. So particularly to do with inflammatory pathways, that we see an upregulation of genes related to anti-inflammatory genes, and a reduction in certain things like interleukin productions that we know are involved in the clearance of these fungal pathogens. In contrast, what we see at the high density, so again this is the less stressed density, we actually see a reduced pathogen immunosuppression. So these fish that are less stressed are better able to mitigate against this sort of immunosuppressive effect this pathogen has. So this was all very interesting to me at the time. You know, of course, we're showing that stocking density is stressed and having an impact on immunity, but there were some other results that were really surprising me. And what I found was in these kind of analyses that we do with gene expression panels, so these what we call gene enrichment tests, so where we're looking to, in these whole big lists of genes that have been changed by a particular treatment, you know, what is the sort of, what's rich in those terms of their functions in these groups. And what I kept getting was these things to do with circadian rhythms. So these are what we call clock genes, and what I found was that both the stocking density treatment and infection itself seem to be quite significantly altering these genes to do with circadian rhythms. So you have here down at the bottom, these are what we call gene co-expression networks. So these are just essentially suites of genes that are sort of being co-expressed together in a similar way across your samples. And then I can overlay the sort of expression changes between healthy and infected fish with sort of more intense colours showing a greater change with infection. And what we can see here is that these more intense red colours is showing that there's more significant changes in these genes happening at these low densities in infected fish. And then we kind of pulled out, because it became very clear that these are all very, very much related to circadian genes. And here this is a panel of all sorts of circadian genes that we know from vertebrates, and they're coloured if they're significantly different between healthy and infected fish. And basically the take-home message from this is that the infection seems to be having a massive impact on these clock genes, and on top of that, the stress level or the density level of these fish was actually exacerbating these changes. So this got me thinking again about what's the relevance of this to aquaculture practices. And actually, interestingly, in fish farms, photoperiod is actually a really commonly used tool for managing fish. So we often manipulate light in fish farms, typically with extending day length, or even in the extreme, holding the fish under constant light to either speed growth or to somehow control their reproduction. And of course, you know, this is done to increase productivity. They want the fish to grow quicker, or they want to stop the fish from going into a reproductive mode and just investing into growth so they can get the maximum sort of meat out of the farms. And also, interestingly, there's now a move towards sort of more indoor farms, so taking these fish farms out of the natural environment and putting them into indoor facilities. And of course, we're then having to make decisions in terms of how we light these sort of highly artificial environments to actually best keep the fish. So I know that there's issues around sort of infection and stress levels on farms that are impacting clocks. We're doing things to photoperiod in fish farms, and this kind of led me to the current research fellowship that is really getting into this: are we exacerbating disease issues with these kind of light manipulation practices? So before I delve into that, I better give just you a quick primer on circadian rhythms and health. So I'm sure you've all heard of circadian or daily rhythms. They're ubiquitous to life, from bacteria and plants and fungi through to us humans. And they're all controlled by these core sets of clock genes, so these endogenous clocks that are being expressed in our bodies that are essentially just transcriptional, translational feedback loops. And here's just a basic vertebrate clock. So we have the period and cryptochrome genes being expressed. Their products build up in the cells. They suppress another pair of genes, the BMAL and clock, which in themselves are promoters of period and cryptochrome. So when period and cryptochrome products build up sufficiently, they block these other genes, which in turn switch the period and cryptochrome genes. And essentially what happens is we just get this rhythmic rise and fall in these gene products throughout day-night cycles. And this leads to a whole suite of rhythmic phenotypes in organisms. And importantly for me, is that this is driving rhythmicity in the immune system. And at least we know from mammals is that our immune systems are incredibly rhythmic. And the sort of broad pattern of rhythms is that in our active periods, so when we're just waking up, our inflammatory responses tend to be most active, and these are sort of our first line of defence against pathogen attack or injury. And then what happens is, as we go into our restful sleep phases, that the other arms of the immune system, the more adaptive immunity genes, tend to kick in to either repair or resolve resolution during our sleep periods. And just as a side note, it's not just free-living organisms that have rhythmicity. Parasites themselves have rhythms, and we know for many, many years things like egg laying, larval emergence and things in parasites are highly rhythmic across daily time courses, but we don't really know whether they have themselves these sort of internal endogenous clocks, or they're just using the cues from the host and the rhythmicity of the host to sort of time what they're doing. So this has become a really sort of revolutionary concept in human medicine in the last couple of years. We now know that humans that have disrupted circadian rhythms, things from jet lag through to shift work, just general bad sleep patterns, can actually increase our risk of disease and things like obesity and susceptibility to cancer. And there's also now a move towards what we're calling chronotherapy, so actually applying treatments, things like chemotherapy, at different times of day to actually increase the efficacy. And in fact, it's thought that things like flu vaccines can be substantially more efficacious if we actually apply it at the right time of day. But no one's really thinking about this in terms of animal health. And that's what I'm really trying to get at with my research fellowship, is looking at some of these big questions. So how does photoperiod contribute to disease issue in fish farms? So by looking at the host rhythmicity, in the rhythmicity of their immune systems under different light periods, whether we can actually see if that has an impact on disease susceptibility. Also, I'd like to look at the rhythmicity of the parasites themselves. So look at how the parasites are responding. Do they have rhythms in their genes? Can we find parasite clock genes? And then finally, I also want to bring in the sort of third component of this research, is looking at the host's microbiome, so their commensal microbiota, and do we see a rhythmicity in these themselves, and whether they actually are interacting with the parasites and the host rhythms, to give us an overall sort of view of the circadian health of these animals. And all of these kind of questions I use are rainbow trout as a model for disease, and their various parasites, and they're actually quite an important fish for, at least in Europe, in terms of aquaculture production. So why look at skin microbiomes? Well, first of all, it's important to note that skin infections are a really, really big problem in fish farms. We have a whole suite of issues with disease, particularly in salmonids with skin diseases. And the skin microbiomes of fish are thought to be really important for disease resistance. They're kind of what we can think of as the first line of defence. In this mucus layer, they provide sort of competition for bad microbes to invade and sort of adhere to the outer surfaces of the fish, and they may even produce sort of antimicrobial compounds that fend off these bad microbes. But it is important to note that there is always some level of sort of pathogenic microbes in fish skin, but they tend to be at very, very low levels unless there's some sort of perturbation of the natural microflora on the fish's skin. So with these kind of experiments, I'm just giving you sort of a broad overview, is we typically raise the fish under sort of different light periods. So here we've got the sort of two extremes. We've got under a sort of very regular 12 hours of light and dark. And then in the other end of the spectrum, we have fish held under constant light. We raise these fish from sort of very small juveniles for a number of months, and then we will separate the fish, and then half of them will be kept under sort of healthy conditions, and the other half are then challenged with these argillus lice. We allow the lice to attach and feed for a period of time, and then we go through circadian sampling. Essentially what we do is try and capture at least 2 full circadian samples, and taking samples of fish, so swabbing them for their microbiome, and taking the fish's skin for gene expression panels, every 4 hours, continuously for 48 hours, which I can tell you is a lot of work. We also have a small subset of fish which we plunge into continuous darkness, which we call free-running conditions, to see if these kind of cycles we're trying to detect continue when we take away the light cues. And that's just a sort of test of true circadian rhythmicity. So we have these DNA swabs for the microbes, and we have the sort of RNA extracts from the fish's skin, and I use 16S metabarcoding to look at the microbial communities using fairly standard pipelines. And then I use sort of direct mRNA detection methods in a specific panel of clock and immune genes using nanostring technology. So then when I've got these microbial community profiles and the gene expression profiles, I can then look at whether these are actually linked to one another. So first of all, what seems to be the impact of photoperiod on these sort of normal measures of aquaculture success in terms of growth? Well, interestingly, when we keep the fish under equal food rations, so these fish are being fed at the same times a day, exactly the same amount, so they have the same access to food. They're just the only difference being that is how long the lights are switched on for. We actually don't really see any difference in growth. But what is really interesting is that when we do challenge them with these Argillus lice, we see a massive difference in their susceptibility. And what we see is that the fish that are held under constant light really struggle to clear any lice off their skin. So they are very, very susceptible to the lice, in contrast to those held under normal light-dark cycles, where we see a really quite good proportion of the lice, even just within a fairly short period of time, cleared off by the fish. So yeah, we're seeing a really big difference in susceptibility just from photoperiod. And then when we look more carefully actually at the immune expression, here we've got a whole suite of different immune genes, from the sort of inflammatory innate genes through to the adaptive immune genes. And don't worry too much about the detail here. What's important to note is that the orange boxes are the Argillus infected fish, and then the green boxes are the healthy fish. And this is just a sort of overall expression response between the two light periods. So we've got the 12-hour light dark and the continuous light. But what became really apparent just looking at this sort of single snapshot was interesting to me was that I'm seeing already some of the biggest upregulations of genes, immune genes in infected fish are ones to do with antimicrobial peptides. So remember, this is a large ectoparasite, so I wouldn't necessarily immediately expect sort of a massive upregulation of things that are usually associated with bacterial infections. But what we always think is that the Argillus infections may be a route of entry for some opportunistic pathogens, and this certainly is kind of a smoking gun for that. And then if you look sort of more broadly, you don't really seem to see a massive difference in the sort of upregulation of these immune genes between the two light periods, but there is some key differences in very interesting genes, in particular these couple of interleukins here at the top. And these two interleukins are thought to be really, really important with clearing crustacean ectoparasites in fish. One of them is to do with sort of initiating a very strong inflammatory response, and the other to do with sort of a call to arms for sort of T cells, which are again sort of very important to sort of resistance in ectoparasites in fish. And it's really interesting that again we see this sort of dampening down of the expression. So yes, they're upregulated in both light periods, but the level of upregulation in constant light is much lower than those in normal light-dark cycles. We also found that we have really strong rhythmicity in the clock genes in fish's skin, which is really interesting. So here we've just got a whole suite of the really core clock genes of vertebrates, and again we have green is healthy, orange is infected, and then we have the solid lines showing if it has significant rhythmicity, and the dots means it's just arrhythmic, we didn't find any rhythm. And immediately what we can see is that there's some really big differences both with infection and light treatment. So here we have a particular gene, BMAL1, and you can see there's significant rhythms in both healthy and infected individuals under light dark, and also in constant light. But you can see the shape of these rhythms have been quite altered, and we can actually look at this statistically. So there's a few measures that we can look at. So there's the mesal, so this is just essentially the average expression level over the rhythm. The phase, so that's basically when is the peak and when is the trough of the rhythm, and then the amplitude is how much is it going up and down, and all of these we can look at as to whether they're significantly different or not. And again, here we have some examples of where we found differences. So for example, you can see down here in clock 3, the healthy fish have a very nice strong rhythm going on. The infected fish under light dark cycles have a slightly dampened down. It's still rhythmic, but the rhythm has been dampened. Contrast that in continuous light, that we see sort of a general chronic upregulation of this gene, so it's rhythmic in healthy fish but at a much higher level, and then rendered completely arrhythmic when challenged with infection. And we see this in a whole load of the clock genes, that the clocks are indeed being rhythmically disrupted with infection. And this is quite interesting because some data is coming out also from a lab that's looking at various fish species under various different ectoparasite challenges of just how active they are through day-night cycles. And what we see is that these fish are indeed much more active when we expect them to be resting when they're challenged with an ectoparasite. And this could be linked back to the fact that their clock genes are being disrupted, so their natural sort of sleep-wake cycles are being disrupted due to infection. So what about the immune genes? Well, we did actually find a significant proportion of the immune genes in the skin were highly rhythmic. And they kind of fit with what we expected, at least from mouse models, in that we see this similar pattern, that we see a great sort of upregulation of the inflammatory. The peaks of the inflammatory responses seem to be coming in the first few hours of light. An increase, sort of the peak of antimicrobial peptide production would appear to be around the sort of middle of their light phases, and then a big shift over to the sort of more adaptive immune genes during their rest or their dark cycles. So things like immunoglobulins and T cells were much more likely to peak during the dark hours than they were in light. And again, you can see from these panels various differences, either due to infection or the light period. So a lot of the immune genes were rendered arrhythmic due to continuous light. A whole suite of things, including the antimicrobial peptides, were no longer found to be rhythmic in continuous light. Also, we found a lot less genes were rhythmic in infected individuals, so only a proportion of the rhythmic genes from healthy fish was also seen to be rhythmic in infected individuals. And those that were rhythmic in both, we often saw a shift in the phase. So the actual timing of when these genes were peaking was being altered either when we take away light cues in continuous light or when they are infected. So it does seem that light conditions and infection can have a very sort of disruptive effect on the molecular clocks and the daily immune expression patterns in fish's skin. So what about the microbiomes? So the two main questions I really wanted to ask from this kind of data is, first of all, just on a simple level, do microbiomes vary by time of day? And then on top of that, are these microbiomes actually being impacted by things like light and infection levels? And here we've just got a sort of basic plot, these alluvial plots, looking at the sort of average abundance of the top sort of most common bacterial species found by time in, again, constant light down in the bottom and a normal light-dark cycle at the top, healthy fish on this side here, and then infected fish over on this side. And the main thing to sort of look at straight away is that is immediately obvious is when we contrast healthy to infected fish, we suddenly see this sort of appearance of much stronger prevalence of certain colours. Certain families of bacteria seem to be quite different and more prevalent in these infected individuals. And we can look at this in more detail here. So here we have a sort of tree of the microbiome of the fish's skin, and then the lineages are being lit up if they're different between the sort of healthy and infected individuals. So again, orange is infected, green is healthy, and we can see there's sort of these bacteroidetes that are more prevalent in infected fish, and then these like bacilli species that seem to be more prevalent in the healthy fish, for example. And again, there's some differences between the sort of light-dark fish and the ones that are held in constant light. But what was really interesting for me was that immediately the most sort of increased taxa that I found in infected fish were ones that were actually associated with pathogenic disease-causing bacteria in fish species. So these here I've just highlighted, particularly the Flavobacterium species, are really strongly associated with these horrific skin diseases that we often see in salmonids. So it was really interesting to actually see that Argillus, an ectoparasite infection, is indeed actually leading to an increase in these pathogenic bacteria in the skin. But what about the timings? Well, we actually saw a really significant proportion of the microbiome seem to be rhythmic in their abundance in the fish's skin. Around 50 genera of bacteria were rhythmic, and here what we have is what we call polar plots. So this you can think of as a clock face, with sort of time going around the clock, and each ball represents a particular bacteria taxa, just coloured by sort of higher taxonomic levels. And where its position is on the clock is when it's most abundant. And you can see there's a whole suite of different bacteria species that are peaking all around the clock. And here's just a couple of plots, so this is just again a bit like the gene expression plots, just plotted out in terms of their abundance. And you can see things that are rhythmic or arrhythmic. So for example, we see in the flavobacteria that it's only in the infected individuals that we see a strong rhythm in their abundance. And they're not actually that particularly rhythmic and at much lower levels in the healthy fish. Interestingly, these again, the bacillus, which we see in the light dark cycles, we have a fairly strong rhythm in their abundance in the fish's skin. But they're almost completely absent when we put them under constant light. And these are actually a really interesting group of bacteria because they're actually often used for probiotic or health promoting in fishes, both guts and their skin. So what's clear from this is that there doesn't really seem to be a particularly temporal pattern, at least at a phylogenetic level. You can see, there's a kind of broad spread of these sort of peaks of bacterial abundance around the clock and no really sort of significant pattern. But we can look at these rhythms in a different way. We can actually look at the rhythms of the functional potential of the microbiome, so what the microbiomes should be or might be what we can predict they're doing at any given time of the day. So here again we have polar plots, but this time the points are actually to do with gene pathways, and these are just broken up into different broad superclasses. So we have sort of biosynthetic here in the blue-greens, degradation and assimilation pathways in the oranges, and sort of energy generation in the purples. And these are just for fish under light dark cycles. And what we can see from this very clearly, there's a very strong temporal pattern to this. So here in the healthy fish, we have in the first few hours of light, the most sort of abundant sort of gene pathways are related to biosynthesis and generation of energy. Contrast that within the first few hours of darkness, we see a lot more degradation pathways. Then contrast that with the infected fish, and then we see a complete sort of skewing of the clock. So everything's happening around 4 hours earlier. So again, now that the sort of biosynthesis and energy generation is happening actually just before the lights come on, so everything's kind of temporally shifted with infection. And then if we add the double whammy of continuous light onto that, we actually see even more disruption. So now we can see some of the peaks are happening still in the healthy fish, but now we're sort of kind of seeing a more spread out, so there's not actually as temporally coordinated. There's kind of now becoming 2 peaks in sort of peak activity levels, and particularly in when they're becoming challenged with infection, biosynthesis becomes really quite spread out and there's no sort of temporal pattern seen in the same kind of way. So we've got this rhythmicity in the immune system of the hosts, we know that, and then we seem to be having some quite strong rhythmicity in the microbial abundance. But is this actually linked? Can we actually begin to link and look at this sort of interface of the host's immune system and their microbiomes and see how that's actually being impacted by infection and light period? So this is where we look at these what we call co-occurrence or association networks, and this is going to be a fairly complicated diagram, so I'll just walk you through this briefly. So what we have here is each ball is either a bacteria species, which or genera represented in these pink dots. Then we have green nodes that are the immune genes, and then the orange nodes are the clock genes of the host. And then the connections are where there's significant associations. So the red is a negative association, and grey lines mean a positive association. And the size of these nodes means their importance in the network. So the bigger the node, the more central or the sort of more keystone they are to this particular network. So here we have the healthy fish under 12-hour light-dark cycles, and what we can see is that the clock genes are really quite central to this sort of interface between the bacterial networks and the host gene expression networks, and they're the sort of cross-linkers that these clock genes are really linking that between the microbes and the host. And so these labels here, actually, I forget to mention, is that the black ones mean they are significantly rhythmic, and if they're white, it means they're arrhythmic. So here again, most of the host's genes are both in immune, and clock genes are arrhythmic, and we can see a whole load of the bacteria are also rhythmic, and there's a links together. So we can then contrast this sort of network topology with those in infected fish, and you can see it becomes suddenly a much more complex picture. So there's a lot more crosstalk going on between the microbial community and the host's expression when there is infection present. But also what's clear now is suddenly we see again this loss of rhythmicity. The clock genes are becoming shifted, they're much smaller, so they're becoming less central to the network. And we see now there's these really big hub genes, and these are sort of T-cell markers, which is interesting. And these seem to be the sort of main drivers that are connecting the microbial community to the host's immune gene expression. And then we can also look under constant light, and here we see what's quite interesting in the healthy fish is that there's much less connectivity going on with regards to the clock genes. So you can see they're much smaller, so again they're less central to the network, so these clock genes aren't playing such a central role in terms of this crosstalk with the microbial community. But again, there's this really sort of strong driver, these T-cell genes. And equally in the infected fish in constant light, we see these T-cell markers are being really important. The clock genes again are not playing such a central role. So what does this all mean? So, well, first of all, this is really the first time looking at a broad spectrum of immune genes and in the fish's skin, and actually showing they have circadian rhythms. And we see that there's peaks of innate immunity during the daytime, the active periods of the fish, with a switch over to sort of more repair and adaptive immunity involved in resolution of infections happening in their restful periods. And what we see is that constant light is actually rendering a lot of these immune rhythms arrhythmic, and that infection itself can actually really perturb the circadian rhythms of the fish and alter the patterns of these rhythms. And it seems to suggest that these are indeed, these sort of long or constant light periods could really be exacerbating disease in aquaculture. Not only that, we found that a sort of a large ectoparasite can massively perturb the commensal skin microbial community in the skin, and that this is first really good evidence that Argus infections are indeed increasing the potential bacterial pathogens to actually enter that community and could be causing these secondary infections. And that the Argus infection again is changing the rhythmicity of the skin bacteria and shifting the phase of the bacterial peaks in the skin. It seems that also that the microbial community of fish is highly temporally organised, at least in terms of what they seem to be doing, and that infection and constant light again could be perturbing these sort of temporally organised functions, and this could have a massive impact for disease issues. But of course we need to dig into this further. These are all at the moment very much association studies. We really need to begin to manipulate these systems to actually see which direction, who's controlling who. If we knock down the microbes, does that have a knock-on effect in terms of gene rhythmicity, or vice versa? But this is indeed the first evidence to suggest that there's a really strong link between fish skin immune activity and their microbial composition, and it seems that both clock genes and T-cell genes could be really these linchpins to sort of guiding the community and what the key players are in that community. So what's this all mean for the future of aquaculture and managed animal health? Well, it's certainly beginning to lay the foundations, I hope, for the utility of chronotherapeutics, so actually timed application of treatments, actually thinking about how we can use chronobiology to manage animal health, and maybe we could actually try and reduce our use of pharmaceuticals, but actually use them in a more efficacious way by actually applying them at time of days when they're going to be most efficient. Not only that, it's just thinking about this issue, these health impacts of constant light, is what does this mean for wild populations? We now know that light pollution is becoming an increasing global issue. More and more natural habitats are being impacted by sky glow, artificial light at night. And we're beginning to see the really detrimental effects of taking away normal light dark cues on immune systems and health. So my question is could this actually be increasing disease risks in wild populations? So I'll leave that there with you to think about. I'd just like to say thank you ever so much to the EMPSEB team for inviting me. I'm glad we're finally getting together to actually share science, and I'm really looking forward to hearing your talks. And I'd just like to leave it there to say thank you ever so much to my colleagues at Cardiff and Aberystwyth universities that helped me with this research, and the amazing genome hubs that do all the cool sequencing for me, and of course my funders, the BBSRC. So thank you very much. Thanks for listening. Bye-bye.